along auditory paths. But these later neurons can be much more selective and say, oh, this is not only auditory information, but this is sound of harmonica played by little Alec, which annoys mother, for example. So those neurons can have much more selective specific information. I try to understand this basic neuronal coding in the cortex, but those the same experimental techniques and data analysis methods can also be used to give us a better understanding of different disorders. And with Bruce McNaughton and multiple other people, we did some experiments where we recorded in epileptic animals, and here you have activity of around 30 neurons recorded in neocortex and hippocampus. And here I'm showing 60 seconds of seizure. So here seizure starts, this seizure happens, and what you can see interestingly is that if you look at this long pattern, those patterns, you see that again there are some specific neurons which are starting seizure, and later other neurons which are only active somewhere in the middle of the seizure. But if we zoom in this very small part of the seizure, what we will find is, so here you have this ictal spikes, and what you can find is that each of those ictal spikes is again composed of some sequences of similar duration as packets. But interestingly, only part of those neurons are somehow entrained to those ictal spikes. There is a lot of other neurons which seems to be completely don't caring about seizure. So those neurons can be recorded on the same electrodes as those neurons, and for some reason it seems like only a portion of neurons are entrained to this global seizing activity. So again, by looking at the same, using the same analysis as for packets, it was possible to discover this sequential activity and that only part of neurons are entrained. And here it was a manuscript by Adam, and I should update, it's not anymore under review. It was just rejected, so if you wrote some great paper but it was rejected by reviewers, then welcome to the club. Okay, so I showed you about those mysterious packet sequences, how those sequences can be generated, how those interconnected network can generate such reproducible patterns. Well, one of the reasons could be, for example, connectivity. So we know that in a brain, neurons are not connected in some random uniform way, but rather there is a lot of weak connections and there is some few stronger connections. So if we assume such architecture, then what we will get is that regardless of stimuli, the activity will tend to propagate through the stronger connections, and depending what was exactly stimuli, there could be some variation on this main pattern. Also, some neurons could be more excitable, so they could be on the beginning of the packets, but it seems like this architecture of the cortex, assuming that there is this non-uniform distribution of strengths of connections, can account for these sequences. And there were some modeling studies which are showing exactly that. So here is some summary picture, and here it is abstract figure. So if here this rectangle would show space of all possible patterns. So you can imagine that each point in this rectangle, it is representing some of the possible patterns. So for example, here it is some sequential pattern, here is kind of sequential, here is kind of completely random pattern. So when we look at the spontaneous activity, we see only some specific subset of patterns observed. Not all possible patterns, but only some subset. And when we present some different stimulus, those stimuli evoke patterns which are not some taken randomly from this space of all possible patterns. Rather, they are taken from this space of spontaneous patterns. So different stimuli will evoke some sequences where the small variation of those sequences, it will be encoding stimuli. And this is something surprising because, you know, you would imagine that from theoretical point of view, to best distinguish this stimulus from this, it would be that responses to this would be somewhere here, responses to other stimuli would be in completely different space. So if they are completely different patterns, then brain should have the easiest way to distinguish those patterns. But for some reason, it seems like it is not how brain works. It seems that all different spontaneous and stimulus patterns are following certain template, and some small variations from this template, it is what is coding stimuli. So this what I was showing you, it was recording in one cortical area, but Majid did some nice imaging study which shows how this activity can spread through the cortex. So here is rat with some visual stimulation. So after 40 milliseconds, you are starting seeing some activity. And what is really interesting is to see how this activity spreads and goes to entire cortex. And it's not only that this activity goes to entire cortex, but this activity also is going back to visual cortex. So what it means is that probably neurons which are active earlier, they are, for example, activated right by stimuli, but neurons which are active later, they are already not only getting information from sensory, about stimulus, but also they are somehow interpreting the stimuli based on this what other brain regions can tell about the stimuli. So now this response can be much more selective. So those patterns, those packets which I was showing you, it seems like it is not just local, it is involving entire cortex, and it helps to integrate information about stimuli. So if there is some stimulus presented, then this information is sent to all the other brain areas. This is interpreted based on your memory, based on your expectation, and for that later this area can have better idea what is the meaning of the stimuli. To somehow put it in a picture, we can imagine the following, that if we, for example, see some cookie jar, then it can evoke some sequence, some packets of activity in visual cortex, so it will be some sequential activity, but this activity will not only stay in visual cortex, but also will propagate to other areas. So it will evoke some, these packets will propagate, for example, to gustatory cortex, and it will tell some information about, recall some information about taste of this cookie. It will generate some sequence. This information will go back to our visual cortex to provide information that this visual stimuli also has this tasty value. And later, if it is confirmed, it can activate some motor behavior, reaching for this cooking and eating. And when we just think about this cookie, it seems like these spontaneous packets are very similar, that we can activate very similar sequential activity. And it is not yet clear, at least to me, what are the differences between evoke and spontaneous. From my analysis, it seems very similar, so it would suggest that we would be living all the time in hallucinations, not distinguishing stimuli, but it is not the case. So somehow there must be some differences between stimulus evoke and spontaneous sequences. Just I don't know yet what are those differences. What tells brain that this is just spontaneous thought rather than actual stimuli. Okay, so I showed you that, you know, in brain when we are actively processing some information, it seems to be this continuous activity, but in fact it is divided in those packets, and that each packet has some sequential structure, that some neurons which are active earlier, have this more broad information, that this is some start of the packet, and later that what exactly this packet is about. So what's big deal about it? So let me give you some analogy. So here it is some this double helix DNA, and it's not enough to know that we have this DNA inside. What is really important is to know how the information is coded here, and to know that this seemingly continuous string is divided in genes, in those packets of information. It has crucial importance to know that, for example, what are the specific sequences which are starting in gene, which are the specific sequences which are terminating gene. So to read this information in a genome, it is in this DNA, it is crucial to know that first we have those genes, and next what are the beginning and end of each gene. And similarly here, to understand what our brain is doing, for example, if we want to in future decode in real time what a rat or human is thinking about, it is really important to know where each packet starts, because informations can be interpreted only in reference to this packet. So if we know how to detect each of those packets and how to interpret information within each packet, it could be equivalent as knowing that this DNA is divided in gene, and knowing what are the markers of beginning and end of each gene. The real summary from this talk is that information in cortex is processed not continuously, but rather in these discrete packets, and each packet has between 50 to a few hundred milliseconds. So it seems like if more complex information is, if stimulus is more difficult to distinguish, then those packets can be longer. If this is just simple stimuli, black and white, it could be shorter duration of those packets. That those sequences for different stimuli are rather common, a variation on one common theme, rather than as people were assuming before that different stimuli will evoke completely different orthogonal patterns. And the function of those packets, it could be to help to integrate information across cortical areas. So by providing brain this few hundred milliseconds of time to process this information, it may help to somehow provide this background information for each stimuli. So if you hear some sound, it may be meaningless for you, but if you heard before that it was a harmonica, you saw this guy playing it, and now it would make much more sense for you how to interpret this sound. And it could be the function of those packets to help to not only take this bottom-up information, but to in this nice way combine with top-down information. And as I said, by knowing that this information is divided in those packets, and knowing what is the structure of those packets, it could really help us to decode what the brain is thinking at any given time. So I think that those packets are those basic building blocks of cortical coding. And many people contributed to this work, and here is just a few. And I got some financial support for this research. And now if you have some questions about this talk or about Krakow, Poland, now is a good time to ask. Thank you, guys.
Yeah, I don't know where the information is in the data that you presented, but just let's play a little mind experiment. If you made a sound up at the front of the room, and then you recorded it from each one of us, each one of us would be active to that sound at a slightly different time. I probably would show activity pretty quickly because I'm close to you, and someone in the back of the room would show activity quite a bit later because they're further away from you. And if you took and you analyzed all that, probably it would tell you where people are in the room, but it won't tell you any kind of intelligent code that of the kind that you seem to be talking about.
So in this analogy, the information what you got, it will be the same as somebody in the back room got. But what I'm saying is that what you will be hearing, it will be just kind of general sound, something, you know, you will not distinguish that this is Polish or English, but the person in the back will able to distinguish every word of what I'm saying. So this is what is the difference between those early and late neurons. Not only that they are receiving earlier information, but if we look to how they respond to different stimuli, it seems like they are responding to any, you know, sound or many sounds, and those later neurons are able to somehow say what exactly the message is about. Does it answer your question, or?
Oh no, I understood your talk, and I understood you said that, but you haven't answered my question, which is just because he's in the back of the room and he answers your response quite late, how do you know that specific information? Like, what's your data for that?
Where is this picture? So, for example, here, you know, by presenting different stimuli, I can see that neurons which are responding earlier, they don't have much information, they are not very specific what tone is presented, but this neuron later is not responding in the same pattern as this to all the tones with just longer latency. It somehow knows much better what tone was presented than this early neuron. So by playing those different tones, different stimuli, and looking how each neuron is responding, I can draw such conclusions. Does it answer your question or still not?
Well, I understand that.
Okay, so maybe you can have this discussion offline, because yeah, I'm interested to. It seems like I'm not understanding your question.
Okay.
Aaron?
So I'm wondering, does the time nature of the packet, does it provide any constraints, or does it suggest anything about, like if you had some time varying stimulus? So if they were too close together, or you know, could the headers and the information interfere with one another? So what happens if, you know, after the initial burst you change the tone, and that's somehow important?
So it is something
starting
what Matt is investigating, this masking, that if this stimulus and mask are, you know, within this short time, in this tens of milliseconds, they can interfere, and it can bias your perception. So it is something what would suggest that those packets can somehow interfere if you are presenting too loud stimuli, and this second can be somehow affected by this first one. Matt?
We found using EEG with humans that there are certain kinds of sounds, especially sounds that have abrupt energy onsets, that create or trigger very early event-related potentials in auditory cortex, and also with very tight time locking. But then other kinds of sounds, for example, when a sound appears abruptly in a different location but had previously been in the scene somewhere else, so that sort of sound isn't accompanied by an energy transient, your brain has to kind of compute that secondarily from spatial information. And those sorts of sounds evoke a signal that's later and more jittered in time, human EEG. You don't see the early events. So I'm curious if you've ever tried using other kinds of stimuli that are not just energy onsets, more complex stimuli that might not have that first tightly time-locked burst, but would only appear as sort of the second packet.
So I would bet one beer that you see this later latency for this sounds is not that they are, if you have some longer packet, and what EEG is doing is basically smoothing this in time, and it would cause this appearance that it has this longer latency. But I would expect that both packets start at the same time, just this more difficult to detect packets without this clear onsets. It requires more processing time, so it is longer, so you will see it kind of more smooth. So for that, this peak, it will be a little bit later. But I would guess that if you would stick some probe to your subject, you will see onset at the same time.
We'd have to give them more than a beer to do that.
Majid?
Yeah, I have two questions for you. So you're talking about neurons, two types of neurons, so early responder versus late responder, and I assume that the data that you have collected either in your lab or you kind of saw it published by other people are mainly collected from the sensory cortex, primary sensory cortex. My question is this being seen also in association areas in the cortex?
So
first question.
First thing, so it is, as I said, it seems like there is continuum, but this I'm showing here, early and late, it's just for simplifications to better explain, but it seems like there is whole continuum of responses. I was also mentioning about those monkey faces, so it was in a IT area where they show something similar with packet organization, so it seems like it is not only constrained to this primary or secondary sensory area, but also in these higher areas you still see this sequential activity.
Okay, but the selectivity, I'm talking about the selectivity.
Selectivity, so again, it was in recording in monkey in IT, and neurons which were firing earlier in this IT had only this information about that this is some face, so if here it would be 5 different faces presented, and this early neuron would be responding to all those faces, but the late neuron in the same area would be responding only to selectively to one specific face.
Okay, now my question is, you are trying to suggest that if we are basically trying, I mean, you're saying that these neurons are, at least the one that you recorded, the primary cortex, and some of these neurons that are responding more selective, but they are getting inputs from some other cortical area, some kind of top-down control or something, right? Is that what you're trying to suggest?
That they can have access to this
Exactly.
information.
Now they are already in a higher association area. Where then they get input? Are they getting also input from some other higher association area?
Thank you for a tricky question. So I would imagine that here those first neurons will be saying in this IT area also that let's prepare there is some information about faces. And those neurons later, there will be some local processing and they will figure out that this is a face of given monkey. So, even in sensory areas, I would imagine that if there is some simple stimuli like here just 2 tones, you don't need that those neurons would be activated by this higher order areas. It seems like this local processing can also has also this dynamic that some neurons are responding earlier, and in this local processing it is figured out that those neurons are responding to this 12 kilohertz. But if there is some more complex stimuli, then this ability of that there is also input from higher order area, it helps to clarify. So I would imagine that for some simple stimuli, easy to distinguish, you don't need really this feedback, but for some more complex, not clear stimuli, it could be beneficial to distinguish.
Okay, my last question.
Maybe let's give, who else has questions?
So my question was about the epilepsy results. Was it that those neurons that are responsive during the ictal events, or those the neurons that are the early responding neurons that are specifically blind, or is it, did you look at that?
Here I didn't look much at spontaneous packets. I mostly look at those packets which are evoked by these seizures, and it seems like most of those early neurons are from hippocampus, and most late neurons are from neocortex, so it will show these locations. But also, even if we look only within hippocampus, on neurons recorded on the same electrode, we will still see that some neurons are consistently active earlier during ictal spikes than other neurons. How it relates to the spontaneous packets, I didn't look enough carefully to give some more information about it.
Is there any way to tease out exactly how much information is encoded within a packet? Like, for example, if you had like a really something you would call like a simple stimuli versus a complex stimuli, would you expect that the simple stimuli would actually be a smaller packet?
So this is what I would expect, that simple stimuli would evoke a shorter packet, that it will be easier to get to the conclusion what the stimuli is exactly, but I didn't test it. Actually, I have some data where we presented this natural sounds, so I would expect that with this natural sounds, those packets would be longer. But one thing, what makes my life difficult is that here it's relatively simple to define where is beginning and where is end. Here, it is very difficult to define where is the beginning and end of each packet. And what I was telling you is based on some statistical analysis. So it means that I averaged, relations between neurons across long period of time, and at single time, I believe that if we would record not tens of neurons but tens of thousands of neurons, then we could clearly see each packet individually and we can address these questions. But with this little information, what we have, probably it would be too difficult.
Do you have views on what part of the packet would be evoking activity in higher order association areas? Or like if the identity of the neurons type 1 or 2, like you described, are evoking activity in the next level up in, say, somatosensory cortex or something like that? Because as you move up a hierarchy, typically you'd get more like higher order feature representations, right? So would the specific firing characteristics then evoke responses in higher areas?
I think that things are quite complex, that you cannot think about it as going just feed forward. There is this continuous looping. So let's say that this spike is propagated not only to other neurons in the same area, but also goes to other areas, and those other areas are now affecting this neuron. So this neuron, it will be slightly differently responding and sending this updated information to this higher order area. So it is not like just forward and backward pass, it is rather like this continuous loop. At least it is my naive thinking about it.
Okay.
Okay guys, so if somebody is interested in more talk, I'm happy to stay and discuss, or feel free to step in my office anytime. Thank you.